All right, let's get right into it. If you're tackling genetics, you know that understanding the nitty-gritty of DNA and RNA structure is, well, everything. So we've put together a super focused guide to help you absolutely nail the core concepts, the stuff that's pretty much guaranteed to pop up on your exam. Let's do this. First things first, let's tackle a question that trips up so many people. Nucleotide versus nucleoside. I know, they sound almost exactly the same, but trust me, that one little difference is a huge deal. Stick with me for just a few minutes, and I promise you'll have this down cold.
Okay, so before we dive into the deep end, we need a roadmap, right? It's so much easier to understand DNA when you think about it in 4 distinct levels. We're going to build it up from the tiniest little piece all the way to the final massive structure. And here they are. It all starts with the nucleotides. Those are our basic building blocks. Then we link them together to form a single strand. From there, 2 of those strands get together and twist up to create that famous double helix. And finally, that helix gets packed and folded super tightly into what we know as chromosomes. We're going to walk through every single one of these steps.
This picture just lays it all out perfectly. See how we can start at the bottom with that big, complex 3-leaf shape and just zoom all the way in, level by level, until we get to the individual letters, the nucleotides, right at the top. It's all about building from simple to complex. So let's start at level 1, the nucleotide. This is the fundamental building block for all nucleic acids. Honestly, if you're in a genetics course, understanding this little guy is totally non-negotiable. Every single nucleotide, doesn't matter if it's for DNA or RNA, is made of 3 simple parts. You've got to commit these to memory: a phosphate group, a 5-carbon sugar, and a base that contains nitrogen. That's it. Just remember, phosphate, sugar, base.
Okay, now check this out, because this is a classic exam question. This is the tiny difference in the sugar that actually gives DNA and RNA their names. Look really close at that number 2 carbon. In RNA's sugar, ribose, you see that little OH group? But in DNA's sugar, it's deoxyribose. That oxygen is gone. Deoxy. It literally means without an oxygen. That single atom, it changes everything.
All right, time to solve our little mystery from the beginning. A nucleoside is just the sugar and the base stuck together. That's it. But the moment you add 1 or 2 or 3 phosphate groups to it, it becomes a nucleotide. So here's the key takeaway. The phosphate is what makes all the difference. And ATP, the cell's energy money, is the perfect example. The adenosine part, that's just the base and the sugar. That's the nucleoside. Then you add 1 phosphate, and boom, you get adenosine monophosphate, a nucleotide. Add another, you get ADP. Add a third, you get ATP. They're all nucleotides, just with a different number of phosphates attached. Okay, so we've totally mastered the building block. Now, let's move up to the next level and see how these individual nucleotides get strung together to build a long, continuous chain. The real magic here is a special kind of bond called a phosphodiester bond. This is a super strong covalent bond, and it's what forms the sugar-phosphate backbone. You can think of this backbone as the 2 long, twisting rails of a ladder. It provides all the structural support, and the bases just hang off into the middle. And here's another concept that is a huge exam favorite: directionality. You have to know this. The phosphodiester bond always connects the 5' carbon of one sugar to the 3' carbon of the very next one. This creates a chain that has a distinct start and finish, a 5' end and a 3' end. This 5' to 3' direction is absolutely fundamental to, well, everything in genetics. So, we have our strand. Now let's grab a second one and build the most famous molecule in all of biology, the DNA double helix. This is where the magic really happens. So how do the 2 strands stick together? With hydrogen bonds between the bases, and the pairing rule is super specific. A always pairs with T, and G always pairs with C. Now, here's a key detail. Notice that AT pairs have 2 hydrogen bonds, but GC pairs have 3. What does that mean? It means GC pairs are stronger. So a stretch of DNA with a lot of G's and C's is going to be more stable and a little tougher to pull apart. Okay, this next part is absolutely 100% crucial. You have to get this. The 2 DNA strands are anti-parallel. It's a term you just got to know for your exam. And it's all about how those 2 strands are lined up next to each other. So what does anti-parallel actually mean? It just means they run in opposite directions. It's like a 2-lane highway.
Take a look at the diagram. You can see the strand on the left goes from 5' at the top down to 3' at the bottom, but its partner on the right, it runs the other way, 3' at the top, 5' at the bottom. This is the only way the whole thing works. The bases just wouldn't line up to form those hydrogen bonds otherwise. So now that we're basically DNA experts, let's go meet its super versatile cousin, RNA, or ribonucleic acid. It plays by a lot of the same rules, but there are a few key plot twists. If you take only one thing away from this section, make it this slide. This is your cheat sheet for the 3 main differences. First, the sugar. DNA's got deoxyribose, RNA's got ribose. Second, the bases. DNA uses thymine, but RNA swaps that out for a similar base called uracil. So in RNA, A pairs with U. And third, the structure. DNA is that stable double helix, while RNA usually hangs out as a single strand. But hey, single-stranded doesn't mean it's just some boring, floppy string. In fact, being single-stranded is RNA's superpower. It means it's incredibly flexible. It can fold back on itself, letting its own bases pair up and create all these cool and complex 3D shapes you see here. Things like bulge loops and that classic stem loop, which kind of looks like a hairpin. And this right here is where structure meets function. This is a model of a transfer RNA, or tRNA. I mean, look at that wild 3D shape. It's not random at all. This very specific folding is what allows it to do its job, which is to read the genetic code and carry the right amino acid to the ribosome. Its shape is its function. And that's our whirlwind tour through the molecular world of DNA and RNA. We started with a simple 3-part nucleotide, and from there we built strands, we built helices, and we saw these incredibly complex functional shapes. Which kind of leaves us with one last big thought. How does this elegant molecular architecture, all governed by just a few simple rules, manage to store and carry out the entire unbelievably complex blueprint for all of life?